Hello everyone, welcome to the next lecture. So today we're going to be looking at complement. So this is probably one of the more complicated lectures in this series. And again, you're not expected to really know every single molecule, every single name, but the most important things I will say just up front will be the three types of pathways and some of the key mediators in each one. So again, here we can just see an example of complement's function. The membrane lesions formed by complement proteins, we see here. We'll talk about those in detail later, but these are directly killing the cell. And here we can just have a brief overview, which I recommend you keep in mind as we go through. So first we'll have our stages of complement active action. So we'll have our pattern recognition trigger. We'll have our protease cascade and the amplification of C3 convertase. And then we'll have our effector functions, which are common to all pathways: inflammation, phagocytosis, and membrane attack. These are the three main functions of complement. So, complement is a family of greater than 30 serum proteins. It is produced in the liver and also circulates in the blood in an inactive precursor state. Complement acts sequentially and it involves pathway amplification. So it kind of goes down in a chain, one molecule to the next. Complement plays many roles. It can lyse bacteria with direct killing through the membrane attack complex, which inserts into the target cell membranes, and it causes transmembrane pores, which kills the gram-negatives. Those are the holes we saw in the cell earlier on the first slide. It also involved in opsonization, which enhances the phagocytosis by bridging pathogen to phagocyte via complement receptor, also known as CR, and also has a role in inflammation through chemoattracting activity. So we can see that complement components deposit on the pathogen surface, which target it for phagocytosis. And we also have the formation of the membrane attack complex, which causes lysis, as we can see right here. So the language of complement. This is probably the trickiest part. It's basically the nomenclature. So as you can see here, a ton of different molecules to learn. This is a good figure to break it down into function as well, but we'll focus on a lot of these in detail later, but this is a good figure just to see function, molecule. But I find it easier to break it down following each pathways, which we'll get to later. So again, there's over 30 complement proteins, and there are also several regulatory proteins which prevent unnecessary host damage because we don't want complement going crazy because it can do a lot of damage. And dealing with so most are named with a C and a number, as we can see here, C3b. And they're named in the order of the discovery, not order of action. So, and also some are cleaved into fragments. So C3 will usually get broken down as C3b, C3a. Same with C2 and C4. Same thing happens. They split in their two B and A. So the A usually refers to the small fragment and B to the large fragment. But the one exception is C2a, which is actually the large fragment, and C2b is a small fragment. And there are also some alternative pathway components which use different letters. So factor B, factor P, BB, BA. We'll get to these later. So probably the most important figure in this lecture, which I recommend, this is the one you probably study the most, is establishing that there are three pathways in complement: alternative, classical, and lectin. And the two innate ones are lectin and alternative, and the adaptive one is the classical pathway because it only acts if antigen-antibody complexes are formed. So you must already have they kind of act in our adaptive immune response. Complement activation is complex and highly regulated, and this prevents accidental activation and causing host damage. So the complement pathway can be activated in three ways. There's the classical pathway, like we said, which requires antibody and is activated during the adaptive response. We have the alternative pathway, which is involved in innate responses, and the lectin pathway, which is in the innate response but uses PRRs. And again, this is just another way to view the three different pathways. So first, we're going to talk about the lectin pathway. So the lectin pathway uses soluble receptors that recognize microbial surfaces to activate the complement cascade. The lectin pathway is activated by mannose, so that's the key thing to keep in mind, via the mannose binding lectin, which is usually referred to as MBL, and also by acetylated sugars in gram-positive walls. This includes N-acetylglucosamine and liponucleic acid, and capsules via phycolins. So phycolin and MBL basically have a similar function, but bind to different things. So this pathway detects differences in terminal glycosylation of glycoproteins on the cell surface of fungi and vertebrates. Yeasts often have a high mannose glycans on the cell surface, and glycan processing in vertebrates produces terminal sialic acid residues instead. So there are 3 ways to activate complement. Again, we're going to look at the lectin pathway right now. So the lectin pathway uses carbohydrate recognition receptors, which are activated by certain repeated microbial patterns. Antibody is not required in this pathway. Otherwise, it is much like the classical pathway. So instead of antibody, it essentially uses the MBL or phycolin, but is still considered innate. So MBL and some phycolins are produced rapidly by the liver during an infection, and these are oligomeric proteins that go from monomers to trimers to oligomers, which increases the avidity, which is the total binding strength. We also have MAS, which form complexes with the MBL and phycolins in the plasma. So MBL and phycolins form complexes with serine proteases, also known as MAS, which stands for mannose binding lectin activated serine proteases. So that's a mouthful. MBL has collagen-like domains and binds to mannose, not sialic acid. And phycolins have fibrinogen-like domains and bind to oligosaccharides with acetylated sugars. So as that can be seen here, we have our phycolins binding to oligosaccharides containing acetylated sugars, and then we're MBL, which binds with high avidity to mannose and fucose residues. Here we can see MAS 1 and 2 interacting with MBL for this action. So MBL or phycolin binds to mannose or acetylated sugars on microbial surface. This binding causes a change in MAS2 conformation, which results in serine protease activity, and this activates the second MAS in the same complex. Activated MAS result in the activation of the complement components. So here we're going to get into some of the more detail. So we can go from C4 to C4A to B, and as we said, most will break down to A to B, B being the bigger, A being the smaller, except for C2. C4B then binds to C3, and MAS cleaves C2 to C2A, C2B, creating C4B2A, which is an active C3 convertase, which is kind of like the main point of convergence between the 3 pathways, but they do have different names in the different pathways. C3 will go to C3B and C3A, forming C4B2A3B, also known as a C5 convertase, and this will deposit and the deposition of C3B on the cell surface. So here we can just see there's the activation of MASP2 associated with the MBL or phycolin. This cleaves the C4, as we discussed, into the two pieces. Then we have C2A change, and again, this is a large one in C2A that binds to the complex. Then we have C3 come in, and then we have our C3B attached, and C3 goes to chemo tract, as you can see here. And then we have a bunch of C3 activation. Even here, we can see up to 1,000 molecules. So again, to summarize the lectin pathway, mannose binding lectin or phycolins bind to carbohydrates and pathogens, and the key mediators are MBL and phycolin, MASP2, C4, and C3. This results in C3 convertase. And then now we have the common pathways. We have C3A and C5e working in inflammation, C3b working in opsonization, and then we have the terminal components we'll get to later forming the membrane attack complex. So again, this is just a reminder. Now we'll be looking at the other pathway. So now we're looking at the classical pathway. So just a brief summary figure here of the proteins of the classical complement pathway. So some things we've seen before. We've seen these before. Again, good reminder to read these, but here we can see the unique ones, C1q, C1r, and C1s. So these complex proteins bind directly to pathogen surfaces or indirectly to antibody bound to pathogens, thus allowing autoactivation of C1r. C1r cleaves C1s to active protease, and while C1s cleaves C4 to C2 and C2. So the classical pathway of complement activation starts only if, this is very important, antibody which is either IgM or IgG, which are the immunoglobulins most used in complement opsonization, is present and bound to an antigen. So this binding opens the C1 binding site, so C1q can then bind to the Fc portion of the antibody, which is essentially, my bad drawing is here at the bottom of the antibody. This activates a sequential complement cascade, ending with production of the membrane attack complex and leading to cell lysis. So again, this is just a more detailed figure of the one figure we saw earlier. So the classical complement pathway of complement activation starts only if antibody is present, as we said. And so here we can just see, here's the antibody molecules binding. So here's our complex, our C1q complex, binding to two antibody molecules, cross-linking. And then we can see here too, then we start the cascade. So we have our C4 breaking down, then our C2 breaking down. Then here we have C3, and so on and so forth. And then eventually at the end, by this point, we kind of follow the same complement pathway. Then we have our membrane attack complex formation. So the classical pathway is initiated by the activation of the C1 complex and is homologous to the lectin pathway in many ways. However, antibody is used for pathogen recognition rather than MPL. And the serine protease in the classical pathway are C1R and C1Q rather than MAS1 and MAS2. So here we can see an electron micrograph, and here's a picture of the C1 complex, where we can see C1R, C1S, and C1Q. So the steps in the classical pathway. So C1Q binds to the Fc portion of the antibody, as we see here. C1Q then binds to serine proteases C1R, C1S, forming the C1QRS. We have C1QRS, which cleaves C4 and C2. So C4 is cleaved, C2 is cleaved, forming C4A, C4B, C2A, and C2B. Then C42BA forms the C3 convertase and releases C3B, as we can see here. Then we have the formation of our C5 convertase, and then our terminal sequence begins. So again, the terminal sequence is going from our C5 convertase to the membrane attack complex. So again, to sum up the classical pathway, we must have antigen-antibody complexes on the pathogen surface. Antibody is a must. Key mediators include C1Q, C1R, C1S, and then C4, C2 again. And then again, we come back to the same pathway of the three divergent functions of complement. So now the alternative pathway. So again, another good figure here, the alternative pathway components. So again, we see similar molecules, but here we can definitely see a change in the nomenclature, factor B, factor D, and properdin, also known as factor P. And this will come in, we'll talk about these in a second. So in the alternative pathway, C3 is the most abundant complement protein. C3 attaches to microbe surfaces and can activate the complement cascade in any of them. Factor B and factor D, factor P, or properdin, interact and cause a stable C3 convertase, which means more C3 can be activated. The C3P complex with factor B forms the C5 convertase, then this leads to our terminal sequence. This activates the next complement protein and triggers the pathway of C5 to C5A to C5B and triggers the cascade of events. So the alternative pathway is activated in two ways. So first, it can be activated through C3B, similar to the other pathways, generated by the other two pathways. So C3B will bind to factor B, cause a conformational change in factor B, which can now be bound by factor D. Factor D cleaves factor B, causing C3BB, forming the C3 convertase and BA. C3BBB is the C3 convertase of the alternative pathway. It has the same role as C4B2A in the classical lectin pathways. So again, you can see in the figure, we have factor B coming in instead of the other molecules with the similar functions in the other two pathways. And then the second way is through by spontaneous hydrolysis, aka tick over. So C3 in water, that's why it gets the term hydrolysis, which occurs ongoing in the plasma, and factor B will bind to this, then factor D will bind, and forming BB and BA. And this will form our C3BB C3 convertase. Again, this is the C3 convertase of the alternate pathway. Basically, this is a spontaneous reaction that occurs as these complement proteins just float freely, not freely, I suppose, but in the blood, compared to targeted pathway like before. So, complement fixation causes the generation of the C3BB and alternative C3 convertase. So, summary of the alternative pathway. This stable C3B through factor B and D on the microbial surface binds factor B and C3B and B complex and binds to factor D. This cleaves factor B to BB and BA, forming the C3BBB or the C3 convertase of the alternate pathway. but C3 convertase is not very stable on its own. So stabilizing an alternative pathway, we need properdin or factor P. So if properdin or factor P binds, this forms the stable C3 convertase that we mentioned earlier. Lots of C3b are on the cell surface, and there's also released C3a fragments. This results in opsonization and chemoattraction. Properdin also binds to microbial surfaces and directs alternative pathway to pathogen surfaces. It also binds damaged mammalian cells, causing C3b deposition, and removal by phagocytosis. Membrane and plasma proteins that regulate the formation and stability of C3 convertases determine the extent of complement activation under different circumstances. Properdin is made by neutrophils, and stored in granules, and is released by activated neutrophils. So that's a key thing to remember. Even though they release all those killing effects, they're also involved in the complement pathway. So again, C3b is the alternative C3 convertase, which generates C3a and C3b, which leads to the pathway amplification we've seen in all the other pathways. So the outcomes of C3 convertase activity. So C3 convertase cleaves more C3 to C3b, and this C3b coat microbes surface with a bunch of C3b. This results in opsonization, while the C3a fragment, which is a small fragment, causes chemoattracts phagocytes. So basically, and a lot of them, this is also similar to C5a, but they act as chemoattractants while others act in binding to the actual cell. So C5 convertase, which is also can be called C3bb P3b in the alternative pathway, results in the terminal events and complement fixation of the membrane attack complex, causing cell lysis. So we have what are called complementary regulatory proteins, which essentially prevent C3BBB, or the C3 convertase, from lysing our own cells. So C3BBB formation on our cell surface is rapidly inactivated by complement regulatory proteins. These regulatory proteins interact with C3b and prevent convertase formation, or they can cause the rapid dissociation of C3 convertase. So example of complement regulatory proteins, or what I think is quite a cool name for a molecule, is decay accelerating factor, DAF, or CD55, and factor H, which compete with factor B for binding to C3b. This causes the dissociation of C3b, and they can also displace BB from the convertase. Factor H has affinity for cellulosic acid, which deactivates C3b on our cell surfaces, and some microbes take advantage of this by having surface cellulosic acid. While factor I, which is a protease, works with MCP, also known as membrane cofactor of proteolysis, can cleave C3b and IC3b, which is the inactivated form. So again, we have our protein stabilizers, which include properdin, which stabilizes C3 convertase. We also have regulators and inactivators, including factor H and factor I. And we also have our DAF and MCP, which can disrupt the process. So again, here's just a summary figure. Just a summary. So here we can see summary of the classical lectin and alternative complement activation pathways, the key activators, initiating complement component, and components common to all pathways. These can all be seen here. Again, good review figure. So again, alternate pathway, C3 undergoes spontaneous hydrolysis to C3H2O to initiate eventual deposition of C3 convertase on microbial surfaces. And here are our key proteins, factor D, B, properdin, and C3. So as we discussed, all the pathways generate a C3 convertase in some way or form, which cleaves C3, leaving C3b bound to the microbial surface and releasing C3a. So the next step in all the pathways is C5 convertase activity. So C3 convertase deposits many C3b fragments on microbial surfaces. The binding of C3b to either of the C3 convertases causes the formation of C5 convertase. In the alternative pathway, this is C3BB plus C3b results in C3b2BB, which is a C5 convertase. Classical and lectin pathways, we have C4b2a3b as a C5 convertase. The result here is that our C5 convertase cleaves C5 into C5a, the small fragment, C5b, the large fragment. C5b then initiates the assembly of the terminal complement components, the membrane attack complex. So here we can see our C3 convertase, or C5 convertase, split, and this results in the activation of the membrane attack complex. we can see here we have our membrane attack cones formed, C9 complement lesion, and the plasma membrane. We'll get to this later, but this is the membrane attack complex. So again, terminal complement components, here are the proteins. So now we have C5, C6, 7, 8, 9. The detail of these is not crucial, but just keep them in mind. One thing to keep in mind is C5a is a mediator of inflammation and is highly active. This is a chemoattractant like C3a. So the assembly of the membrane attack complex. So essentially this is much easier to follow as it just goes from C5, sorry for that writing, C5 to C9. So we can see C5b binds to C6, C7, then this binds via C7 to the complex. Then we have C8 join, and then we have a bunch, 10 to 16 molecules of C9 bind and form the pore. So this is also another point for regulating complement activity. So on the cell surface, we can see we have this membrane attack complex. They would form normally, but we have the molecule C59, CD59 on humans, whereas pathogens don't have C59. This binds to C5b, 6, 7, 8, and this complex prevents recruitment of C9 to form the pore. So CD59, aka protectin, which is probably easier to remember, in the host cell membranes is intrinsic protein that inhibits C9 binding. So here we can kind of get a nice picture of the entire pathway of the membrane attack complex. So again, the key actions were opsonization, phagocytosis, inducing inflammation, and the membrane attack complex. So there was the three. So again, certain complement proteins act as opsonins, enhancing phagocytosis. So for example, a bacteria coated with C3b, when only C3b binds to CR1, bacteria are not phagocytosed. But C5a can activate macrophages to phagocytose via CR1. So opsonization is a key action of complement. The complement receptors, or the CRs, bind complement components attached to pathogens and promotes phagocytosis. For example, CR1 binds to C3b. Opsonization requires synergy between complement components. C5a binds to the C5a receptor and activates macrophages to phagocytose bacteria bound to CR1. C5a binding to C5a receptor causes signal transduction and phagocyte activation. So I know a lot of the focus is on C3b, and C5b, but we must not forget about the little A ones, especially C3a and C5a. So here we can see certain small fragments like C5a, C3a, and C4a contribute to inflammatory responses. These increase the vascular permeability and blood flow. They can chemoattract neutrophils. They can also increase the permeability, which allows for fluid and protein accumulation, which results in traffic to lymph nodes and facilitates adaptive responses. So they have several functions outside of. They basically work with the adaptive immune system and our other cells. The activation of phagocytes, including macrophages and neutrophils, also promotes pathogen clearance. So we can see here, all these molecules go to the vessel, cause increased permeability, which allows for increased fluid leakage from the blood vessels and extravasation of immunoglobulin. And we also have the migration of macrophages through chemoattractant activity. So here we can see C5a induces histamine release from basophils and mast cells, which causes increased vascular permeability and inflammation. It can also chemoattract neutrophils, macrophages, and lymphocytes. And the role in histamine release by mast cells and basophils causes increased vascular permeability, allowing for cells to pass through the vessel easier and allowing for increased extravasation. Again, complement plays a key role in inflammation. Neutrophils and monocytes and lymphocytes leave the circulation through extravasation, and they enter the tissues that participate in the immune response. Complement allows us to be as efficient as possible and allows us to be very targeted. So again, complement proteins play several roles in immunity. We have the membrane attack complex, which forms pores in bacterial cell membranes, causing cell lysis. We have the inflammatory effects and chemoattractic activity, where certain small fragments like C3a and C5a can chemoattract neutrophils and induce histamine release from basophils and mast cells, and causing increased vascular permeability. And we also have opsonization, where C3b is an opsonin and facilitates the phagocytosis via phagocyte complement receptors. Again, see you next time. Thanks for listening. Take care.